Thank you, Alberto. Good morning. First of all, thanks to the organizer and for Marco Montorsi for the kind invitation. I'm kind of strange here because I'm a scientist, I'm an immunologist, and I work upstairs in the Department of Immunology and Inflammation. And I'm here to talk about the links between inflammation and cancer. And I will briefly touch at the end some of the latest results that we have obtained in studying inflammatory mediators in pancreatic cancer development and progression. So to start, already in 2004, the magazine Time dedicated a cover to the problem of inflammation as an underlying mechanism of several human diseases, including cancer. And when I'm talking about inflammation, I always mean chronic inflammation, persistent inflammation over time, because the acute inflammation, which is the host response of the innate immunity, is usually self-resolving and is important to face pathogens or to face tissue injury and repair. But the chronic inflammation is the one that is giving problems. So which are the links between cancer development and inflammation? Epidemiological studies have clearly demonstrated that conditions of chronic inflammation predispose to carcinogenesis, at least in selected tissues. The other important epidemiological evidence is that the long-term use of non-steroidal anti-inflammatory agents decrease the risk of some cancer, like gastrointestinal colorectal cancer. Then we have some more cell biology evidence, the one that we study. And the evidence is that in most tumors, it is present in the microenvironment, the expression of several inflammatory mediators like inflammatory cytokines, chemokines, proteolytic enzyme, and so on, and also inflammatory cells, mainly macrophages. And we will briefly discuss on these mediators. And the other evidence, and there are several more, in fact, but I choose only a few, in experimental mouse cancer model, the deletion or inhibition of specific inflammatory mediators, like, for instance, TNF or IL-6, decrease the development of cancer. And there are a few examples with studies with short numbers of patients, also in human, that specific polymorphism for inflammatory cytokines or mediators, in fact, protect against the risk of cancer. So we have inflammation as a risk of carcinogenesis, and we have an inflammatory microenvironment also in cancers which are not related to inflammation. So how to put together the two things? We can say that two pathway links cancer and inflammation. This, of course, is a schematic representation. And we have an extrinsic pathway when inflammation comes from outside, like in inflammatory condition, in infections. Here is a list of several pathogens that all of us know, virus, bacteria, HBV, papillomavirus, chronic infections with Helicobacter pylori. They are all linked to persistent inflammation and occurrence of carcinogenesis in the target tissue. There are also autoimmune disease or autoinflammatory disease where chronic inflammation, like in IBD or respiratory disease, can promote the development of cancer. So this is inflammation coming from outside, specifically linked to pathogens, but not only, also to irritants or tissue injury. And then there is an intrinsic pathway, which is intrinsic to the neoplastic cells, which are the oncogenetic events occurring in tumors. And these are, for instance, activation of oncogenes, like RAS in pancreatic cancer and other, RET-PTC in thyroid cancers, and several other, or mutation of tumor suppressor, like p53. So genetic mutations that occurs in neoplastic cells are also able to trigger chronic inflammation. And so both pathways converge to the same effect. And Schematically, the effect is the activation of the inflammatory cascade, so activation of transcription factors, mainly NFKB, but also others. Production of inflammatory mediators like inflammatory cytokines, chemokines, and so on. Recruitment at the tumor site of inflammatory cells, and so cancer-related inflammation, which impact on tumor cell progression, because these soluble mediators are growth factors that increase the proliferation and the survival of tumor cells, the epithelial to mesenchymal transition, the switch on of angiogenesis, mobility of tumor cells invasion and metastasis, and last but not least, inhibition of adaptive immunity. So in general, cancer-related inflammation promotes tumor progression. And I will briefly mention a few concepts on inflammatory cells, mainly macrophages, and on chemokines, which is one of the topics that we are studying. So inflammatory cells are present abundantly in most tumors. Up to 40–50% of the stromal, of the total cells in the stroma are leukocytes, mainly lymphocytes and macrophages, also other cells, neutrophils, but most of them are macrophages, which are immune cells deputed to defend the body against the attack of pathogens. But in fact, the concept that has emerged in the last 10 or more years, also boosted from our research group, is the concept that macrophages are a very plastic, heterogeneous population, which differentiate from the precursor, the circulating monocytes. And we can, here again, schematically divide an M1 type macrophage, which is able to phagocytose bacteria, to kill the bacteria, also tumor cells, to stimulate the immune response. An M2 type macrophage, which is in fact able to dampen the immune response, so is immune suppressive. And in the tumor, because this is the type that is most frequently found in tumors, is producing growth factor, which will aid tumor cells to survive, to proliferate, to invade. Macrophages are most important for the switch on of angiogenesis, and in fact they are quite responsible for the failure of chemotherapy and hormone therapy. So the take-home message is that M1 macrophages are important for resistance to pathogens, to activate the immune response, although in chronic situations they can also provoke tissue injury, and they have tumor resistance. But in the tumor, this is the type of macrophage that is most frequent, and they are able to do tumor promoting, although in homeostasis, this type of macrophages are important for wound healing and for angiogenesis, as well as for embryogenesis. And to convince you that I'm not joking, this is a New England Journal of Medicine paper in March this month, in which they have found that the density of infiltrating tumor-associated macrophages in Hodgkin lymphoma is correlated with poor survival, treatment failure, shorter progression-free survival, and disease-specific survival and resistance to relapse after autologous bone marrow transplantation. So the reactive cells in the stroma of tumors, mainly macrophages, are really important for the progression of tumors. Now, the second topic I want to introduce are chemokines, also because our research data are on this field. And chemokines are small proteins, are small cytokines, which are produced by several cells and induce the mobilization of other cells chemotactically along a chemotactic gradient. And the field of chemokines, which were discovered 25 years ago, and cancers have always been strictly connected, because tumor cells produce chemokines, and these chemokines recruit leukocytes from the circulation, for instance, monocytes, because macrophages are so important for tumor cells that tumor cells secrete cytokines that selectively recruit monocytes to their own advantage. And so the major function of chemokines is to regulate the tissue trafficking of leukocytes. But in the last 10 years, it had become clear that also other epithelial type cells have chemokine receptors, not only leukocytes. So chemokines are not only regulating the influx of inflammatory cells in acute inflammation, but also in other situations. And tumor cells may respond to chemokine receptors. And besides migration, which is of course important for tumor cells, other important functions of chemokines are, for instance, the regulation of proliferation and, above all, resistance to apoptosis, or survival of tumor cells, which may be important at distant sites. Another important point is the activation of matrix proteases, or tissue invasion. And so chemokines are very important for tumor cells. This is a brief and recent overview that we depicted for a review, in which different chemokine receptors have been discovered on tumor cells. They bind different ligands, they are specific ligands, and are involved in the invasion and site-specific metastatization of neoplastic cells, like, for instance, CCR7 to lymph nodes and also to brain, CXCR4 to lungs, and so on. So we, years ago, started to have an interest in pancreatic cancer from the cell biology point of view. And what I'm going to present are an overview of our results on the chemokine system in pancreatic cancer. This is the work of Federica Marchesi, now a postdoc in our group. And the work was also done in collaboration with the Hospital San Raffaele. We started to do a profiling of chemokine receptor expression in pancreatic cancer, human ductal pancreatic carcinoma. These are the different chemokine receptors with different cell lines. You see CXCR4, which is in fact the most frequently expressed in all tumors, was present, and we did an old work with this. And then we found that an unusual receptor was expressed in several cell lines, CX3CR1. And this was quite intriguing because at the time, only leukocytes were expressing the receptor, not this type of receptor, not epithelial cells. This is to show you how complex is the family of chemokines. It's a huge redundant family with 50 ligands or more, 19 receptors, 7 transmembrane receptors. You see there is frequently a lot of promiscuity. Many ligands bind the same receptor, but the CX3R1 receptor binds only one ligand. And this ligand is a chemokine which is quite peculiar because is one example, unique example of a chemokine which is bound to the membrane. So in fact, can act as an adhesion molecule because it's bound to the membrane. Can also be shedded, cleaved by enzymes, and is like just other chemokines. Now, which are the producers? The producers are endothelial cells and neural cells, neurons, but also in the ganglia along the peripheral nerves, and usually after activation with inflammatory cytokines like TNF. So the fact that CX3L1, which is also called neurotactin or fractalkine, was produced by neural cells triggered us the idea that maybe pancreatic cancer cells expressing the receptor could recognize and adhere to the ligand expressed on neuron cells. So our research observation was connected to the clinical observation that our clinicians taught us, that pancreatic cancer has a strong tropism for peripheral nerves and ganglia. So it is not infrequent, actually it's very frequent if one looks at it, to find carcinoma cell inside the perineural space. And although not consistently checked, there are a number of studies demonstrating that perineural invasion is frequent and is usually associated with bad prognosis and local tumor recurrence. And I did a a PubMed research, and I found that, in fact, many other tumor types have perineural invasion, most of all prostate, bladder, and others. So it's quite a frequent route of dissemination. So our hypothesis was that the expression of this receptor was important for pancreatic tumor cells to adhere to peripheral nerves. So first of all, we did some functional studies with the tumor cell lines, and we demonstrated that there is more adhesion on cells expressing spontaneously the receptor or a cell line that we have created to express the receptor. On endothelial cells and on neural cells, we use a neuroblastoma to mimic neural cells. And we also molecularly identified that this adhesion occurs by activation of beta-1 integrins and focal adhesion kinase. But what's important is to go to the patients. So the pathologist checked a tissue macroarray of 60 samples, 60 resected pancreatic cancer, and they did immunohistochemistry for the receptor, and the carcinoma were stained intensely, and while the normal pancreas was negative, was very faintly positive. This is the distribution of the receptor expression in the tissue macroarray. Few patients were negative, and most patients were positive with moderate or weak or intense positivity. What is important to observe is that half of the patients were positive, and actually most of the cells, up from 60 to 100%, were positive. So the pathologist scored the intensity of receptor expression, and then they correlated this information with the frequency of perineural tropism, which means that they went back to the original paraffin block and checked the number of peripheral nerves that were engaged by infiltration of tumor cells. And we put it together, all the results, which are here. Neurotropism, 6 is the highest score of receptor, so the higher the receptor is, more frequent is the neural invasion, and this was significantly different. And they also, the clinicians also checked the local recurrence of the disease in this cohort of patients. What is clear is that if the expression of the receptor is high, the frequency of local recurrence is significantly higher. Also, it was checked the time of local recurrence, which is earlier in patients with high receptor expression, and the overall survival was not significantly different. So doing Cox and logistic significance, it turned out that the high receptor expression was significantly associated with the local recurrence of the disease. And no other parameters were in fact associated with local recurrence. Also radicality, it was not significant, and I know that this is an important parameter. And by Cox regression, the perineural invasion was associated with local recurrence. Also, with the multivariate analysis, the receptor score was significantly associated, so it's an independent prognostic factor for local recurrence in pancreatic cancer. So next, and these are data from Luigi Laghi, collaborating all together in this institute. This is another cohort of patients, pancreatic carcinoma, 61 patients. Luigi also checked ampullary tumors, endocrine tumors, mucinous tumors, and interestingly, ampullary tumors and endocrine tumors are negative for the expression of the receptor, but he perfectly confirmed the high expression of the receptor in ductal carcinoma. The important question is why this tumor upregulated this receptor, and is it an early event? So Luigi and Paolo and other colleagues performed an immunohistochemistry on preneoplastic lesion, and this is 3 examples of PANIN in different grade. And you see PANIN 1 already have, in a minority of the cases, expression of the receptor, and this expression increased with the grade of PANIN. So the take-home message is that already pre-neoplastic lesions upregulate the CXC3R1 expression. And finally, and this is a very recent analysis that Luigi did, selecting those patients that were not advanced with small tumors and zero, of course, no metastasis. These are only 26 patients, so these are preliminary data that we have to reinforce. But the proof of concept is that patients that have high receptor expression, in fact, have a lower overall survival. So in conclusion, the function of chemokine receptor expression in pancreatic cancer, this fractal kine receptor, we demonstrated that the receptor is expressed in tumors, but not in epithelial normal cells or very low expressed. It's functional. I didn't show data on migration, but importantly, mediates the adhesion of tumor cells to other cells expressing the ligand, and I show you neural cells, but also endothelial cells of the vessels are, of course important for angioinvasion. In a human study, receptor expression correlates with perineural tropism and is an independent prognostic factor for early local recurrence in resected patients. And finally, receptor expression is found also in pre-neoplastic lesion. So the work was a long-standing collaboration between the two institutes, the Clinical Institute Humanitas and the Scientific Institute in San Raffaele. Federica and Lorenzo are the major person doing all the basic and some of the clinical job, and I want to thank the pathologists, the clinicians, the surgeons. Mantovani is our scientific director, and also in San Raffaele Institute, Alessandro Zerbi, which shifted here, and Professor Di Carlo. Thank you for the attention.